Good time of day, colleagues. My name is Kukuruza, and today I would like to present the results of my bioinformatical analysis titled Classification and Functional Analysis of Gene Expression Dynamics in the Human Placenta During Uncomplicated Pregnancy. First and foremost, I would like to express my sincere gratitude to Maria Blanska, head of the Systems Biology Group in the Institute of Molecular Biology of National Academy of Sciences of Ukraine, professor and doctor of biological sciences, for scientific counseling throughout the process of conducting this work. Placenta is a temporary organ that forms as a part of the embryo-placenta mother system during pregnancy. Its main functions are acting as a barrier between both organisms, supporting their mutual tolerance, facilitating nutrient, oxygen, CO2, and waste product exchange. Gene expression controls how the placenta develops and its function, and thus impacts the health of the baby and the mother throughout gestation and in the following life. Most studies compare gene expression between two time points of placenta's development. Others use a case control comparison, which is mainly done at term. However, gene expression dynamics during the whole gestation period are poorly investigated. Such knowledge is crucial for detecting critical time points and specific stages of pregnancy for diagnostics, predictions of complications, and their treatment. Moreover, it is known that similar expression dynamics may encompass genes sharing biological functions. Clustering genes according to their gene expression thus may give a new insight into the interpretation of placental gene expression during gestation. This study aimed to find the variance, if any, of coherently operating genes in the human placenta during uncomplicated pregnancy through applying the method of dynamical clustering. For this study, placental gene expression data was extracted from the gene expression omnibus database, from the microarray data obtained from healthy individuals, from the series with the IDs that you can see on the slide. The data was then normalized and combined into time series with 4 data points, which are the intervals from the 1st to 12th week of gestation, from the 13th to 20th week of gestation, from the 21st to 27th week of gestation, from 28th to 36th week of gestation, and from 37th week of gestation to the end of gestation. However, there was no data available for the period of 21st to 27th week, so this time point was missing in the further analysis. The average values of gene expression in each interval were calculated, and then differentially expressed genes were identified based on the expression data and used in the clustering using the Dirichlet Gaussian process method, which jointly models cluster numbers via the Dirichlet process and temporal dependencies through the Gaussian process. As an example of how the Gaussian process models the dynamic process and the advantages of this modeling, you can see the image in the slide showing the results of the modeling a time series data through Gaussian process. In the picture on the left, the black dot is the data which is not used in the calculations at that point, and you can see we obtain a graph which consists of the line of the parameter changing throughout time, and then we also get the area of standard deviation in gray, which shows how confident we are about the shape of the graph, meaning that on the right side of the A picture you can see that since there is no data around that time period, the graph, the standard deviation is quite wide, and we are not quite confident how the parameter changes through time. However, once we add this data point in, and you can see on the picture B, the results are that we are now we could predict better how the dynamics changes, and the standard deviation area also changes, showing that we are more confident around the obtained point, but still unconfident around the time point of 4, where we don't have any data. Based on the described analysis, a list of 369 differentially expressed genes was identified. They were used in clustering, and then 9 unique clusters were obtained, each containing genes with similar expression dynamics across gestation. Here on the next slide, you can see the 9 clusters. Each cluster is represented via the graph of the gene expression dynamics, which was unique to it. I also obtained the posterior similarity matrix, which shows the probability of a pair of genes belonging to the same cluster, with white meaning a high probability, close to 100%, while black means close to 0% of genes belonging to the same cluster. Here you can see that all 9 clusters are mainly white, meaning that the clusters were robust, and throughout different iterations of the clustering, they mostly did not change and contained the same genes. Um, Here, once again, you can see the dynamic change of gene expression, with the line in the middle showing the mean, while the blue area shows the standard deviation around each time point. As you can see, in some of the clusters, there's quite high uncertainty around the period of 20th to 27th weeks of pregnancy, because, as I described, we did not have this data point. Based on the analysis of each of these 9 clusters, it was found that each cluster had a mirror pair, which allowed us to separate the clusters into 4 groups. For example, if you look at the group 1, you can see that it contains clusters 1 and 9, because the gene expression in cluster 1 starts high, then it suddenly decreases and stays around the same level after the second trimester of pregnancy, while in the cluster 9 the opposite happens. The gene expression increases and stays around the same level around the second trimester. The same can be said about groups 2, 3, and 4. However, group 2 had had cluster number 2, which had 2 mirror clusters, to clusters 8 and 6, which are very similar in shape. Um, the gene lists that were obtained were then used in overrepresentation analysis using the FCA package in the environment R, and terms from gene ontology aspect of biological processes. And the results of the overrepresentation analysis were plotted using the tree plot function from the enrich plot package, which allows us also to group the biological pathways by the genes that partook in their enrichment, as you can see on the left, meaning that similar gene lists in pathways were grouped together, allowing us to generalize the results even more. And they were also used to build the protein-protein interaction network. This was done in the cytoscape environment using the data from string app database and our results. Here you can see each node represents a gene with either blue or red area or aura around them. This aura, being red or blue, represents which of the polar opposites of dynamics this gene belongs to. As you can see on the next slide, we have the group 1, which contained mirrored clusters mirrored to each other, clusters 1 and 9. There are indicated on the right of the slide with blue or red outline, which is also represented on the protein-protein interaction network, with the nodes that have a red outline having the dynamics of their gene expression being shown in the bottom right as the graph with the red outline, and the proteins which have the blue outline around them, the changes in gene expression of the genes that encode this protein are shown in the top right of the slide with the blue outline. We also looked, once again, as I said, at the overrepresentation analysis, and the generalized results here are shown on the slide, with also the various colors assigned to different biological pathways that were enriched in group 1. And on the protein interaction network, you can see that the inside of which node, which represents the protein encoded by a certain gene, there are also the same colors which were given to the biological processes. and if a node has a particular color inside it, it means that um, it partook in the enrichment of the biological pathways which belong to this group. Um, Overall, the group 1 was enriched in pathways that referred to such processes such as immune response, cellular defense against chemical agents such as metal ions, uh, maintenance of the physiological stability and barrier functions, tissue organization, tissue and organ development, hormone metabolism, and endocrine regulation. Group 2 was enriched in pathways which referred to as cytokine, chemical, and lipid responses, uh, innate immune regulation and microbial components, immune defense mechanism against pathogens, indicating a role in cell signaling and inflammatory stimuli regulation. Uh, Group 2 was also enriched in chemotaxis and uh, leukocyte migration, and broader processes of coordination and regulation of adaptive immunity. This group contained clusters 2, 6, and 8, which you can see on the right side of the slide, expression of which sharply changes from the first to second trimester and then drastically rebounds afterwards, rising or falling respectively by preterm. Group 3 was enriched in biological pathways that referred to hormone regulation, uh, lipoprotein and triglyceride and steroid metabolism, lipid localization, cholesterol transport, vascular medicated transport, and phagocytosis regulation, as well as general reproductive and multicellular organismal processes. Um, Group 2 contained clusters 3 and 4, in which the expression of genes either slowly rises or decreases throughout pregnancy. Group 4 was enriched in biological pathways that referred to development and morphogenesis of the heart and its valves, mesenchymal transformations and the issue development events, regulatory mechanism acting as systemic and on systemic and organismal levels, as well as intracellular signaling pathways, cellular mobility and proliferation, including immune signaling and movement. Group 4 contained clusters 4 and 5, which once again you can see on the right side of the slide, which represented genes whose expression only moderately changed from the first trimester to second, but then drastically increased or decreased by preterm. As you can see, based on the protein-protein interaction networks shown on each slide, there is high amount of interaction between the identified genes in each cluster, or I mean within the proteins which are coded by each gene, uh, especially between groups of different expression. So between the red and the blue aura genes, uh, meaning that genes expression of which rises and genes expression of which decreases during the same time point were interacting and partaking together in the processes which were found to be enriched. It was also found that the functional enrichment was not specific to only the red genes or the blue genes, meaning the one or the polar opposite of the gene expression, and the um, each of the biological processes with both regulated by the both of the by the gene expression changes in the both of the mirrored clusters. This brings me to our conclusions. So the defined variants of gene expression dynamics, which are represented by 9 clusters, function simultaneously during physiological development of the human placenta, and they can be grouped into 4 distinct groups of mirror types of gene expression dynamics. This, together with the results of our representation analysis and obtained protein-protein interaction networks, allows us to suggest that said genes can, in the obtained groups, synergistically or antagonistically partake in processes such as immune system regulation, maintenance of the physiological stability, cellular defense against toxins and pathogens, dynamic immune signaling, regulation of metabolism and reproductive processes, developmental and structural mechanisms. Thank you for your attention.